Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about salt bridges and we are going to choose correct statement. So what are the salt bridges? Take a look at this picture and you see here are groups of amino acid lysine, which is positively charged, and our group of the glutamic acid, which is negatively charged, and we see here interaction which is electrostatic interactions between two R groups with different charge. But also we see here hydrogen bonding. So what is a salt bridge? It is a combination of the electrostatic interactions and hydrogen bonding. So salt bridges are a combination of both electrostatic interactions and hydrogen bonding. Today we have to choose not the only one correct statement, but two. So let's check. Salt bridges form when oppositely charged amino acid side chains contact each other. This is correct statement, but let's find another one. And variant B form when positively charged amino acid side chains contact each other. This is wrong statement. They have to be oppositely charged. Variant C form mostly in the core of the molecule, and right away the next statement form mostly on the surface of the molecule. So which one is correct? Take a look. Here is the globular protein, and as you know, inside of the globular protein mostly hydrophobic and outside is hydrophilic. This also means that most charged R groups of amino acid we can find outside of the protein on the exterior of the protein. It doesn't mean that we cannot find them inside, but the statistics are as follows. Charged R groups would be 4:1 exterior to interior. Per every 4 charged R groups which we can find in exterior, this is only one which we can find in the interior. What also interesting that interior of the globular proteins is dielectric, hydrophobic, and R groups which are oppositely charged would make much stronger connections between them than those which we can find on the exterior of the protein, and those which we can find in the interior. Their bonding almost as strong as covalent bonding. And those salt bridges that we can find on the exterior of the protein can be broken by, for example, change in the pH. In order for salt bridge to form, the distance between R groups have to be no more than 4 angstroms, and in this case salt bridge can form. But if the distance is going to be more than 4 angstroms, then such salt bridge is not going to be formed. So as you see, we have another correct answer, which is answer D. Salt bridges form mostly on the surface of the molecule. It doesn't mean that we cannot find salt bridges inside, in the interior of the protein, but the ratio is going to be 4 on the exterior to 1 in the interior. Per 40 on the exterior, we are going to find 10 inside of the protein. But of course you should understand that this ratio can change from one protein to another one. This is just on the average. So scientists compared many proteins and have found that if you stretch them and calculate number of amino acids per every 150 amino acids, on the average we can find 4 salt bridges on the exterior of the protein and 1 in the interior of the protein. I hope you have learned something new today, and this is all for today. See you in the next video. Goodbye.